Okay, so today we are going to talk about enzymes. Okay, you have learned enzymes from biology, right? So who is going to tell me what are enzymes? Do you have any idea what enzymes are?
Substances that
Yes,
are
Amy?
used. Okay, substances that are used to speed up a reaction.
Yes. So enzymes work in what kind of reactions?
They are biological molecules.
Bravo.
Yes,
Okay.
they speed up the rate of chemical reaction.
Okay, super. So this picture, what shows to you? Do you have any idea what these are, these chains? Okay, let's answer. Let's see the keywords. So enzymes, substrate, enzyme substrate complex, biological molecule, biological catalyst, cell, and protein. Okay, you should think about what are they. Amabel just talked about, and Dami also. Do you have any idea how they work? Do they have specific functions? Do they need specific conditions to work? Do they work alone? So these are the questions you should think about. So watching this picture tells the mechanisms of enzyme activity, okay? So the first thing shows the substrate in the enzyme. So the enzyme has a specific substrate. So then it is created enzyme substrate complex, okay? So the enzyme modify the substrate in order to release products then, okay? So this is how pretty much is the mechanism of the enzyme. Okay, just a second. Here. So enzymes are proteins that act as biological catalysts. Catalysts accelerate chemical reactions. The molecules upon which enzymes may act are called substrates, and the enzyme converts the substrates into different molecules known as products. So here it is the mechanism of the enzyme activity. So the enzymes recognize its substrate, then the enzymes bind and modifies the substrate, then the enzyme releases the products. So what are enzymes? Are biological molecules, typically proteins, that significantly speed up the rate of virtually all the chemical reactions that take place within cells, so within the living organisms. They are vital for life and serve a wide range of important functions in the body, such as aiding in digestion and metabolism. Okay, so again, they are biological catalysts, lower the activation energy for the reaction inside our body, lower the activation energy, and fasten the rate of the reaction. They speed up the reaction by lowering, well, the activation energy. Let's see a video together where we can see how the enzymes work. Okay, just a moment, please. Okay.
Single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur.
So basically lower the activation energy and the products are formed faster, okay? So this is the minimum energy required for the reaction to take place.
To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form an intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell, in every living creature on the planet.
Okay, so pretty much this was how the enzyme works. So what works? What you saw is an example from the Krebs cycle. So aconitase, it's called the enzyme that turns the citrate into isocitrate. Okay, so how it works is like this. So the citrate from the aconitase create the intermediate phase, so cis-aconitase, then aconitase puts the group H and OH into different places, creating isocitrate. So what is that shows is the substrate plus enzymes and gives us the product, okay, in different words. Citrate plus aconitase gives isocitrate. Properties of enzymes are pH sensitivity, catalytic property, reversibility, specificity, heat sensitivity. So you should remember these properties of enzymes. So they do not work when they do not have the proper conditions, okay? So factors affecting the enzymatic activity are temperature, water, pH, substrate, enzymes, inhibitors, end products, activators, and radiation. These are the factors that affect the enzymatic activity. So in conclusions, enzymes are biological catalysts which speed up the rate of different chemical reactions that happens in our body by lowering their activation energy. Enzymes have specific functions and bind with their specific substrates, which are determined by their function to create the substrate complex. Some of the enzymes are amylase, lactase, protease, lipase, ligase, polymerase, ATPase, catalase, nuclease, etc. Okay, as you saw, all ends with ASE. Okay, so can you answer to these questions? As we just talked about, what are enzymes? Again, who is going to answer? Yes, Amabel? What are enzymes?
No, and answers.
Yes?
So enzymes are biological molecules that speed up the rate of a chemical reaction.
Which is the mechanism of enzymes activity? What you should remember? Okay, let's, yes, Fiona, yes, please. As you can see from the picture, so which is the mechanism of the enzyme activity? So what the enzyme does? Yes, Alvin? Come on. So looking at the picture, as you can see on the screen, so which is the enzyme's activity? So the first step is the enzymes recognize its substrate, okay? So this is the enzyme and recognizes substrate. What it does then?
Enzyme binds and modifies the substrate.
Okay, modifies in order to a specific function. Then what is that? What is that?
And enzyme releases the products.
Okay, it's that simple, okay? You're not going into details for that, just in this way you are going to explain it. So which are the factors affecting enzymatic activity? Yes, Alvin, you again? Which are the factors we just talked about? Which are the factors we talked about?
There's αντιθέμον.
Yes, you can help, Alvin, yes?
Amabel.
Yes.
Temperature, water, pH.
Good job.
Inhibitors, I think, and products. Radiation.
Concentration.
Activators.
Okay. So we need to have specific, let's say, environment or conditions in order for the enzymes to work, to work in the living organisms. Okay, which is the other question? Which are the properties of enzymes? What does the enzymes? So we talk about here, okay? They are reversible, okay? They are specific. They have heat sensitivity, okay? pH sensitivity and catalytic property. So they are specific, do not forget that. Okay, so for this lesson, you have to answer to the question, what is a biological catalyst and what is an enzyme substrate complex? So these are the questions you are going to answer to your homework notebook.